Hello, my name is Sarah Campbell. I am a sophomore and I am very excited to present to you a project that took me over 2.5 months to do about earthworms and their effect on the decomposition of plastic. So I first wanted to start this project for two reasons. One, I have always been interested in worms ever since I was a young kid. And two, I have always been a big advocate in trying to deal with the worldwide issue of plastic. As you know, we are struggling with plastic because it is very hard to decompose. And I want to ask you this, will there ever be a world without plastic? I know that we're trying to find replacements to plastic, such as reusable bags or metal straws. But even if we find replacements, there will always probably be objects with plastic in them, or there will always be mounds of plastic still on the side. And the reason plastic does not decompose as quickly as other natural materials is because of a human-made chemical found in it called polyethylene, which allows plastic to stay the same way it is for thousands of years. So before this project, I researched articles and science projects based off of worms and plastic together. And another project used mealworms and had bacteria in their stomachs, which enabled them to decompose the plastic. And this is different from mine, as I am instead using earthworms, and my worms are from a breeder, therefore they are in an enclosed environment far away from this certain bacteria. Another study I also researched demonstrated and explained how earthworms can act differently based on their breed, where they are from, and that can influence how they react with each other, what they eat, and just the soil type around them can influence a lot about a worm. And these studies helped lead me to my question and problem that can the invertebrates known as earthworms be able to decompose certain types of plastic once they are placed in the plastic environment? And my hypothesis after researching this is yes. If earthworms are placed in the same environment as plastic, they will be able to decompose the plastic. So for this project, I first used 3 bins and I drilled holes in the bottom of each so that when we pour water in there to keep the soil moist, the water disperses evenly and it does not drown out the worms at the bottom. For my control variable bin with no plastic, I just mixed in soil with natural materials. And then for the second bin, I used styrofoam balls, broke them up and spread the pieces and mixed those in with the soil as well. And then for my third bin, I used plastic beads and I poured those in and I mixed those with the soil as the first layer. And then the second layer for all of them is a layer of cardboard. Instead of one flat piece of cardboard, I broke up the cardboard in order for the worms to keep moving through each layer as they please. I also used a scale, a pH soil tester, and a measuring tape and ruler to help study and measure the worms throughout this period to collect my data. The pH soil tester was mostly used to study other factors that may influence the results, such as pH, temperature, and moisture content within. I also used sifters at the end to help find and sort out the plastic to measure for a difference in the end as well. This is a summary of what I was explaining to you earlier. And here are some pictures demonstrating my project. So here we have my project all laid out with the first layer. The bottom is none, the second is styrofoam, and the third on top is the plastic beads. As you can see in the plastic bins, there is green, that is referring to lettuce. I did put natural materials in both the plastic, as I did not know how the earthworms will react with the plastic and to keep the experiment consistent. So I took pictures post data collecting of all the worms, and here are some. So here are the worms in the plastic beads bin, here are the worms in the styrofoam bin, and here are the worms in the nut bin. It's important to know that when I'm collecting my data, I chose 11 worms at random from each bin each week, and I masked them, and then I measured their length separately. So this table represents the lengths of the average of 11 worms for each week. So since they were all measured individually, the worms length was then averaged from each bin, and it is very good to see that all the worms did grow in the end. Their length became bigger in all of the categories and bins, and in the end you could see that the none bin did the best, meanwhile the styrofoam did not. Here is the average, the mass on average, and as you can see, this also successfully proved that the worms were able to grow at the end of the study. And again, it is shown that none did well, or the regular compost bin did well, meanwhile styrofoam did not, as well as the plastic beads did very well on this as well. So the difference in the amount of materials from each bin, so this is representing the plastic, so this is not applicable to the none bin, and in the styrofoam and plastic beads we did find a difference, therefore proving decomposition. As you can see from first glance, it looks like plastic beads has the most decomposed plastic, but due to density and proportional wise, styrofoam actually had the most decomposed compared to the plastic beads bin. So here are the difference in the number of earthworms to see how the worms did do. So as you can see, both the none and the plastic beads did not have as many worms deceased or missing than the styrofoam bin. As you can see, this is a visual representation of table 2 with the mass. In week 1, there is a huge outlier for all of the bins because there was an issue with the scale and for it not to be on a flat surface, therefore reading inaccurately. This graph is a visual representation of table 1 with the lengths, and the outlier in week 2 for the regular compost or the none bin was when the worms extend to their full length, and was not like the other bins whose worms stayed pretty short and didn't extend to their full length. This is a summary of what I was discussing with my results as I went over earlier. I have 3 variables throughout. I have the control variable of the none bin without plastic in it. I had the independent variable of the plastic environment, changing what plastic was in it, such as the none, styrofoam, and the plastic beads. And then I had the dependent variable of the worms, measuring how they did in response to that. So as I mentioned earlier, I measured other factors such as pH and temperature. pH and temperature were seen to be proportionally related, as they were in a separate graph that I did place them in, and as one went up, so did the other, as one went down, so did the other. So in this project, no clear pattern was identified with the results, therefore meaning that a longer study period would be needed in order to prove so. And also it is important to know that when I was measuring length, I went to the nearest ½ inch, therefore to not alter anything and to keep it consistent throughout. In conclusion, my hypothesis was supported by the data. The earthworms were also able to grow a little bit in the end. Comparing plastic beads in the styrofoam bin, the plastic beads, like the styrofoam, seem to not do as well in the length category, but in comparison to styrofoam, plastic beads was a little bit heavier and mass-wise. Styrofoam did decompose more plastic than plastic beads, and that may have had a negative impact on the worms, as since they decompose more plastic, that's why they may have suffered in both the mass and the length category. So in the none bin was known beforehand that the none bin would probably do the best, and it did. Referring to other studies at the beginning, I researched a study and it said that the texture at the bottom of the bin, if it has a grainy-like texture, that means that you reached the final stage of composting. And in the end, when I was sorting out using the sifters to find the plastic, that grainy texture was reached. Therefore, this experiment successfully reached the last stage of compost. The different layers of transport, so this is referring to the different sizes of materials such as the styrofoam. So the larger pieces of styrofoam were at the bottom, while the middle-sized pieces were in the middle, and the smaller pieces were at the top. And this was actually in a study beforehand saying that these pieces would be where they were. Also, the study was able to successfully reproduce worms, and this may have had an impact on my study as styrofoams had the most reproduction and showed the first signs of reproduction. So since the 11 worms were chosen at random, that means that the smaller worms could be chosen other than the larger worms in that bin, therefore altering the results. During the study, I mostly fed fruit to the worms, and they seemed to appeal mostly to bananas. So recommendations for the study later on. So if I were to redo the study again, I would not do it in the winter, as next time I would like to make sure I had an outside source of water like a hose to find the plastic better in the end and to make it more accurate. I also want to not have it in the winter, as this could have impacted the worms with the temperature being so low, and I had them in my garage, and that could have made them slow and unable to move as quickly, and therefore not have them decompose as much. Also, I want to make sure I have a specific breed next time. Even though I got these from a breeder, no specific breed was given. Therefore, that could alter my results, as the study beforehand showed that different breeds can alter what the worms do and what their behavior is. In the future, if I were to do this again, I would also dissect the worms to physically prove that the plastic was decomposed. There could be other reasons why the plastic was missing or gone, or why there was a difference, as plastic could have fallen out through the holes at the bottom. The plastic could have fallen off the worms when measuring them and finding the data, and there could also be acidity in the soil, as like I said, proportionally, since temperature and pH are related, and since it was winter and cold, the pH was lower and could have had acidity in that way. The next step in this project to further hypothesis would be to study other plastic materials. So since it's easily breakable and light, I would study other plastics like it, such as water bottles, as that is flimsy. I would also extend this hypothesis and see if other plants can come off of this. So since a grainy texture was reached at the bottom of the bin, I want to be sure and see if other organisms can live off this environment that the worms proved while decomposing plastic, and to see if I can relate that to this world issue today. Here are all my sources that I use, and I'd like to take this time to acknowledge my family for helping me with this project every night and counting worms. And I want to thank my science teacher for always being there and helping me create this study for the way it is today. Thank you for listening. Have a wonderful day.